squamous cell carcinoma of the head and neck.
Good afternoon. Hi, my name is David Neal Hayes. I'm a medical oncologist from the University of North Carolina Lineberger Comprehensive Cancer Center, and I'll be talking to you about the comprehensive genome characterization of squamous cell carcinoma of the head and neck. Just to put this project into perspective, there are 20 tumor types studied by the Cancer Genome Atlas. This is, I think, the 8th one to come forward. The first one, the glioblastoma, which was first reported in 2008, has been cited in over 2,000 manuscripts since its publication. That's an enormous number of citations, and I can tell you that squamous cell carcinoma of the head and neck data set is much, much bigger than that. So this is a very big project. I need to comment that I'm presenting on behalf of both the patients who really motivate us in this work, but also my co-chairs, Dr. Adele El-Nagar and Jennifer Grandis from MD Anderson and the University of Pittsburgh, respectively, as well as the hundreds of researchers and faculty associated with the project. Squamous cell carcinoma of the head and neck is a common disease. It's the 5th most common cancer worldwide. It's number 6 in the United States, with 45,000 cases per year in the United States. This is a disease which is overwhelmingly associated with smoking, an 80% attributable risk. But very interestingly, in recent years, has been associated with an epidemic of the human papillomavirus, and that biology is reasonably well known and described in the cartoon on the right. I want to start with the 10,000-foot view of squamous cell carcinoma of the head and neck, and what I mean by that is I'm showing the cancer genomes of 279 patients, the entire cancer genome of those patients, and the way to read this figure is that patients are shown in the columns, so there are 279 columns. There are 22 rows representing the human chromosomes, and at the intersection of a column and a row is a pixel. It's either white, blue, or red. If the pixel is white, that means that the patient has the normal amount of DNA at that position in the genome, and that would be similar to a normal cell. If the pixel is blue, then that means that chromosomal material or genes have been lost. If it's red, then there are extra copies of the genome at that position. And what's amazing about this figure is that if you look across patients who are completely independent, there is an incredible pressure for these tumors to acquire a very specific set of losses and gains. And you'll see this as the blue and the red stripes across these 279 patients. And in fact, and this is one of the observations from the Cancer Genome Atlas, I think it's a real contribution. If you actually look across different tumor types, so this is squamous cell carcinoma of the lung, squamous cell carcinoma of the head and neck, including HPV positive and HPV negative tumors, as well as cervical cancer, so diseases that are very distant from each other in the body. However, they've assumed a very similar set of gains and losses across these chromosomes, suggesting that there is the possibility to start looking at cancer across tumor types as molecular patterns that can be leveraged and understood both for biologic and therapeutic standpoints. Now, that's in contrast to some other tumors. So here, outlined in red is ovarian cancer, outlined in green is glioblastoma, and you'll see very different patterns of gains and losses suggesting very different abnormalities in these cancers. So moving from 10,000 feet all the way down to ground level, what I'm now showing are mutations or individual alterations in individual genes. The list that I'm showing you here is the 18 most significantly mutated genes in head and neck cancer. And the importance of these genes is, from a statistical standpoint at least, these must be the most important genes in head and neck cancer because they're the most frequent. These are the driving genes behind the cancer, and we really need to understand these. What the Cancer Genome Atlas has shown here, not only are there a number of new genes and novel mutations, but we've also drastically redefined some of the rates of mutations. So I'll just point out the mutation TP53, the historical mutation rate somewhere around 44%, that's shown on the left, documented by the Cosmic Catalog of Mutations, compared to 72%, which was seen in the Cancer Genome Atlas. So that's a pretty big difference. And those differences are important, and as we put together mutation data in combinations of mutations and as well as other patient information, and I'm showing you that in this figure, again, patients are in the columns and the genes or alterations are in the rows. And if you look at the patterns that emerge, I think some interesting things can be seen. Number one, you can see that for the patients who are in red on the top row, who are HPV infected, they almost never have a TP53 mutation. So that pattern is interesting. It's not necessarily therapeutically actionable because TP53 is not a druggable gene. But what it does show us is that there are a few patients who are HPV negative who are P53 wild type. So that means that they have no P53 mutation. And that starts to point us to ask questions like, what's different about these patients? And using the Cancer Genome Atlas data, where we have very high-quality mutation data, we're able to look and see that, in fact, many of those patients have a mutation in a very druggable gene, HRAS, and that's shown on the bottom row here. Many of these previous concepts are somewhat theoretical concepts. As we're engaged in this type of project, we're very cognizant that we need to bring therapies and actionable events forward as quickly as possible, so we also mine this data for the candidate therapeutic targets. And in the interest of time, I'll just point out two of them to you. Starting on the right side of the figure, if you look in the HPV positive patients under the green column, you'll see that really there are almost no actionable events, no actionable mutations in the HPV positive patients, except for one, PIK3CA mutation, which occurs in almost 40% of the HPV positive samples. So that's a very near-term actionable observation. In the HPV negative samples, there's a large number of mutations, including some that are high frequency, including EGFR, the fibroblast growth factor receptors, and CCND1, which could be acted upon in the relative short term for patients. Now, not every patient has a druggable mutation, so that pushes us to look not just at mutations, but also at pathways. The details of this figure aren't necessarily important, other than to say we can put these head and neck cancer pathway mutations into pathways and suggest which pathways might be most productive for targeting at downstream events. And finally, just a comment about all of this complexity. It seems like we've made a lot of observations, but we also made things much more complicated, which can in itself be a great challenge. But these data also allow us to recognize patterns and simplify things somewhat. And what I'm showing here are gene expression subtypes. Again, patients in the columns, genes in the rows. If we use this gene expression data, we can define 4 subtypes of head and neck cancer, not hundreds of subtypes, but 4 subtypes that might be, and these subtypes might be leveraged in terms of the pathways that are altered and the therapies that might be offered for patient benefit. So with that, I'll just conclude with a photo of our, this is just part of the analysis team that met at the Lineberger Comprehensive Cancer Center in September of this year. These folks are all there because they care about these genomic data and understand the genomics of head and neck cancer, and this is part of the product of the Cancer Genome Atlas, is bringing together all these disparate researchers from many different institutions. So with that, I'll conclude.